through interdisciplinary lenses. The speaker will be Professor Lau Pun Chuan. He's the Deputy Head of the Department of Biological Sciences at NUS, and he's a member of the Mechanobiology Institute. He's recognized for discovering VCH protein domains that are involved in the regulation of the growth, the death, the differentiation, the migration of cells. Pun Chuan, please.
Sure. Well, thank you so much, Lim Sung, for your kind introductions, and a very good day for all the audience and my colleagues here. So I'm actually very delighted to be given this opportunity to share with you our very exciting scientific journey of discovery leading towards better understanding of diseases. Of course, the kind of title seems a bit scary, right, when things go wrong. But in our research, basically, we're also trying to compare and contrast what would be normal cell signaling in the normal physiology, and hoping that when you compare contrast, you'll figure out what went wrong. So as earlier on, and Prof. Lupin and the rest of the discussion, science indeed is interdisciplinary. So today, although I'm actually a trained biochemist and practice as a molecular cell biologist, you will see that lots of a very exciting discovery have been supported, nurtured, and collaborated through the whole great teams of scientists, engineers within the NUS. So I kind of start with these few so-called the properties in mind: why sizes, shapes, even the speeds of molecular interactions and trafficking define the functions. What is so important, how they can be controlled, and what went wrong, how they went wrong, and how can we understand the basis of what they went wrong, so that we can, at least somehow we can prevent, if not curing the disease at some point, right? So this is a kind of overarching motivation for much of our work within the NUS. So my humble objective today is very simple. I hope that students, can walk away from my talk to have a greater appreciation of the use of state-of-the-art technologies, the strategy and approach that we use, interdisciplinarity, collaborations through the various expertise, and more importantly, you can see how we're able to address some of the fundamental principle or basic problem, and then try to implicate or translate into some possible application. And throughout the objective 2, 3, 4, and 5, you probably can appreciate, I will touch on a little bit about the cell signaling. This is a cell signaling 101, and where we will engage some principle of protein-protein interactions, and also how uncontrolled signaling due to the defective protein-protein interaction can lead to diseases. And hopefully, this is where we come in to exploit the knowledge. Now, of course, we are very blessed, right? We've gone through these, stages of running development, where we are. We have been blessed with the fact that every single biological processes go or went well. But of course, these things have the process going on in the body. We don't know until a certain disease or states appear. So therefore, we keep a lookout for every single step of biological process. So here I'm just kind of like sum out in these three kind of like different aspects of the biological processes, from the cells and tissue organization during the embryonic development, to the way that even some of the cardiac stem cell niche that can be starting to create new specialized lineages like cardiomyocytes, smooth muscle, and endothelial cells, or the neuronal differentiation, just to name a few. But certainly, things did went wrong, and unfortunately, those are unfortunate cases where you can see some of the very drastic development defects. And if we could understand exactly what had gone wrong, I think definitely there will be a lot of things we can do for the humankind. Now, certainly there are more incidences that nowadays, heart disease is the major killer. In fact, second part of is going to be devoted to some of the most recent study in the heart, study of heart proliferation differentiation. And when the size does matter, when you have a larger heart, it doesn't mean you're going to have a better capacity to live longer and so on and so forth. It turned out to be opposite. And likewise, there is a whole bunch of work to be going on in the field where scientists try to look at the biochemical principle of cancer biology. But I think later on, you'll see that much of those questions nowadays have been addressed using interdisciplinary approach, especially in the new emerging field that we call mechanobiology. So this will give a context, and as a cell and tissue move about, and it's not just about single kind of like chemical constituents, they talk to each other. They involve a lot of more this collaborative between the tissues, among the tissues. Just give a few. Even for the cancer cells start to rip apart, start to move about, they form a different shapes, different size, different mobility, different speed, right? So I have a better cartoon to show you actually. This is one of the review articles that we wrote with my students together with my colleagues, Siti Lim from the engineering. We are looking at the way when the tumor is growing, they're subject to a lot of these mechanical influences, right? The 3D environment from compressive stress to metric stiffness, stretching, topography, all the way until the cells starting to rip off from the tumor. They're starting to migrate out. They are going to penetrate through the endothelial cells, the process we call internalizations, and it's starting to swim around even to this period of time. They are going to find a niche, all right, a place where they're starting to colonize. But doing so, of course, a lot of physical and biochemical process starting to take on. So the entire field of mechanobiology or tumorigenesis and metastasis indeed is a very hot topic to move in. I just use it as one of the example, right? And this is one of the reasons why I kind of thinking science indeed is interdisciplinary. Now, just to give you a context, in fact, it's nothing kind of like, reason. It's been years when scientists started to realize that when you start to plate the mesenchymal stem cells on the different stiffness, the soft stiffness, they become a soft tissue like brain. You put them on a stiffness, kind of like a substrate, they become bone cell. All this can be created without much chemical influences. So this is where a lot of excitement going on to understand exactly how cells sense, how cells transduce the mechanical embracing signal into the nucleus to tell cell what to do with it. In fact, this is one of the driving principles where some 15 years ago, we started this mechanobiology institute, within the NUS context. So we thank for the kind of like interdisciplinary approach with great colleagues to actually work on this field. So for us, within the lab, you can see from here, kind of like clockwise, this is a pretty much like a roadmap, for most of the thing we do in my lab. We are starting to use molecular, genetic, biochemical, biophysical, in fact, also structural biology to start looking at how the protein work, right? And also what they do. And nowadays, we do see the bioimaging, some of which, like what probably been mentioned just now, very kind of like, exciting, kind of like for being able to actually locate and also to understand real lifetime how this process takes place. So this is in a very subcellular level. This is at a very molecular level, and this case is cellular level. You can see the cancer cells are very penetrative, very invasive. We go through endothelial cell. And at the same time, you also see how some of the molecular complex can be trafficking, right? And also for this part, you're looking at some of the polarized cell migration using biosensor. Of course, as a biologist, we ought to be able to relate underlying processes and what the consequence in the tissue and organs. And we'll be using some of the patient samples, some mice model, even using zebrafish is one example. So all in all, I think you can see from here, our approach have to be well integrated. And this is always a challenge, but I wanted to encourage you to think about it. And overarching theme is that we believe that whatever the molecule talk to one another, they will require some kind of a scaffold protein, right? The scaffold protein is there in order to make the thing work and to define the size, shape, and the speed. And all these scaffold protein will actually kind of sense or relate to the biochemical and physical kind of signals from outside and to inside. So the quick question is like this, right? So we are actually motivated by the fact that there will be, and I believe there will be more scaffold proteins that integrate molecule in the right place at the right time so that they all can allow their shuttling, transport, and modification leading to function, or their effective mechanism leads to some other pathology. So lo and behold, some more than 20 years ago, and I discovered, and my lab subsequently pioneered, much of the categorization of the VOH domain is a very straightforward name, kind of like defined through two proteins, VNIC2 and CD4 gap, for which one region which is shared or highly conserved. And when the different protein in the genome, right, started to have a very similar kind of feature, you started to wonder what they are doing there. They had to be indeed important, otherwise they would be conserved throughout the entire evolution. Indeed, the VOH domain have been well defined. It's now have been characterized as a scaffold. They will bring lots of exciting molecular switch, and some of which I don't have time to go into. Suffice to say that they are important in integrating much of the molecular switch in the cells, providing cell dynamic and cell metabolism. You can see from here, genome analysis, cross-species analysis, bioinformatics, at least point out the VOH domain actually define or evolve much earlier on from this evolution. Okay, so before I go into the most recent work of the heart, the biology, let me just put up 2 or 3 different findings First of all, this is about the neurological disorder called ataxia. You can see that individuals suffering from a mutation for this molecule VOH, which is one of the molecule that we work on. Down here, the brain-specific one, and we actually identified this VOH using BC domain can actually carry cargo. What are these cargo? These are the enzymes responsible for the production of the neurotransmitter, the acetylcholine, one of the earliest neurotransmitter ever discovered, in order to send the signal to the synapse or send back the signal in order for the neuron to grow. You can see from here, we even use zebrafish. This is a larvae of zebrafish. We knock down the gene, the larvae fail to respond to the environment. It just simply cannot jump up and move about. When you replace the loss of gene from the zebrafish using the mice version, you can see that now the larvae are very happily jumping about. And this is a mutant on the mice that cannot stand by itself. So at least we now find out the mechanism is that there's a mutation in the VOH that failed to actually bring the cargo out that lead to the disease. And then we also have other VOH protein that cause cancer. Right here, suffice to say, this is where some of the important, for those of you who might know a little bit about kinases, they are enzymes responsible for modifying some of the key substrate, making them change their shape, change their property, change their function. Right? Just this group of VOH protein actually causing pro-oncogenic. It means in the normal cell growth, they are important, they are fine. But when their expression is elevated or loss function, they become it can lead to cancer causing. Or in this case, they even can cause a tumor and also metastasis of tumor. So without going into detail, it is interesting that you can see that how the different VOH domain actually confer different function within different genes and different proteins. We're getting near to where we will be talking soon about the cardiac biology. But again, there's another molecule which will be target of today's talk. This guy we need to actually express quite ubiquitously in all the tissue, right? This is important molecule expressed in many tissues. And you can see from here is this highly expressed also in smooth muscle, heart muscle, skeletal muscle. And years ago, in collaboration with colleagues from the Mount Sinai in New York, we were able to show that this molecule is important for muscle differentiation. In most recent time, in the Science Advance paper, we showed that it actually suppressed cancer cell migration. So it worked like a brake. It actually kind of slow down the migration. In normal circumstances, it's important, right? But in the tumor, when this expression is lost, in fact, we can see that VNIP2 is lowly expressed. It means its expression is low in many forms of cancer. So the brake is lost and the tumor becomes metastatic. And we recently, in collaboration with my colleague from the Department of Biological Sciences, we solved the exchange structure and published PNAS. We showed that you actually have a very unique regional protein we call motif that bind to one of the molecular switch. So that leads us to a most recent paper, and so that is a cardiac disease. This is a major killer. I won't go too much into actually the detail, but just about to say that how we approach it, what we found. In fact, this is a cardiac CVD disease. Actually, it's more incident, more major killer than the cancer that we can think of. Although this data is coming from the United States, it's actually applicable to many countries as well. So the problem with this, right? I mean, there's a lot of heart disease, whether or not it's going to be potentially repairable. And scientists and bioengineers have been thinking a lot how we can go back and refine this, rewiring this, can we fine tune it so that we can repair a broken heart, so we speak. But unfortunately not, because when the animals starting to become adult, the problem here is they lost capacity to grow. But if it's embryo, yes, no problem, they can proliferate. But once it's starting to be more neonatal, unfortunately, the scarring comes back as an attempt to repair some of the damage. So the problem is really partly we can be addressed, because when the proliferation capacity is reduced, when the adult in the embryo grow to adulthood, the inhibitory signals starting to appear. There are lots of signals. For some reasons, they actually come back to prevent this heart cell from proliferating, right? And more importantly, for as far as mechanobiology is concerned, you can see that the cardiac myocyte differentiation actually takes on the stiffness. But however, there's a sweet spot. Right, you can see there's a sweet spot. If you have too much stiffness, the differentiation also cannot proceed. So in the final part of my talk here is going to give a bit of like a background. We use the H9C2 blast harvested from the left ventricular of the rat, and we use it because of various properties, kind of like similar to, but not exactly for the heart cell, but it's good enough as a model to start off with. And suffice to say that we knock down the VNIP2, the heart differentiation is gone. All these are the so-called the biomarker for the heart development. The knockdown actually gone. You can see the biomarker for them no longer exists. And interestingly, to understand how and why VNIP2 important for controlling differentiation, we use a knockdown nowadays, molecular biologists love to use this CRISPR or RNAi or interferon RNA, knock it down. You can see that the whole bunch of genes suddenly pop up. And these are genes responsible for the growth. And lo and behold, it turned out to be YAP. This is a very hot area. This is a transcription co-activator. They will drive many genes important for proliferation. So basically like this, when BD2 is there, it actually suppresses growth. It promotes differentiation. When BD2 is not there, all the important genes for growth starting to pop up. And from there, we ask why. So using the mass spec proteomic approach, we identified various partners, and one of which is a kinase. And it turns out that not only this LAT, which is important as a kinase, the phosphorylated YAP, and not only that, all the genes got turned on, we believe that actually YAP and LATs are now brought together by BD2 as a scaffold. You can see from here, all the imaging analysis really confirm such an important phenomena. Likewise, using biochemical interactions, you can see that BD2 actually interact with LATs and as well as the other partner. So this field context is very important for those of you coming through the field of the cell signaling. You got to know what is a YAP. It's a mechanism-sensitive transcription co-activator. If the cells are plated in a very sparse, the cell need to grow, this factor will go into the nucleus, turn on genes for growth. If the cell grow on very stiff substrate, they will also grow. However, in contrast, if the cell are the soft substrate, they cannot use a fully density, they cannot grow much. Of course, this is the feature of normal cell. Right? In the cancerous cell, all this unfortunately, all this inhibition process is lost. So that's why the cancer cell keep on growing. All right, just to give a context, this is exactly what happened when the cell sense the rigidity, the YAP will drag into the nucleus. Now, for a long time, the scientists believe that proliferation and chiral mass differentiation are coupled together, where this YAP go into the nucleus. It turns out that we proved that this is wrong. They indeed believe to actually sit at the cross section between the proliferation and chiral mass. And this is where we found. So to cut long story short, we found out that BD2 is a scaffold that bridge this kinase and YAP together, phosphorylating YAP, preventing them going to the nucleus, cannot turn on the gene. And for some reason that we still do know, that now all the growth genes turn off. And then in contrast, all the differentiation genes starting to come out. And this is what currently. Now, one last part of the slide I want to show you here. In fact, our investigation pick up a lot of interdisciplinarity as well. You're using the traction force microscopy by actually coding the bits on some of the pillars on the substrate. We're able to kind of monitor their migration pattern and come out with the kind of estimation of a good force. You can see from here, we need to not only allow the YAPs and LATs come together, it does so requiring the force generated through another pathway that we don't have time to go to. And using the laser ablation, we're able to see that when BD2 is there, the contractility is there, the cytoskeleton, which is some part of the cell structure that provide the structural integrity, is very highly contractile. So when you use a laser to actually cut it out, you can see that there is a huge recoil. So all in all, I'm very happy that this paper just been published maybe a month or 2 months ago. The first online version is out. The full version comes very soon. So at the mechanical level, you can see from here, BD2 will engage in another pathway I don't have time to describe. They're then creating a highly cellular contractility that for some reason that we still yet to investigate, that these complexes come together, preventing them from going to the nucleus. So BD2 now is defined as a new mechanomodulator. It is published in the Run Science. So where are we? Now, of course, as the biochemists and the biologists, we interface with a lot of exciting colleagues in the NUS, and many of us working towards using some of the pluripotent stem cell in order to re-engineer some of the cardiac tissue for various reasons. Either you do for drug screening, to look at the regenerative medicine, and so on and so forth. So where is BD2? So we believe that we are now providing a new anchor, a new target, where this molecule as a scaffold actually sit at the cross section between proliferation and differentiation. So I guess this is a very nice cartoon AI generated picture from my colleague Diego NBI. We are preparing this as one of the inside front cover for the journal. And all in all, the last slide here basically telling you that we are not done. In fact, this is beginning a new project as to how, as a result of contractibility, that this complex is brought together. All right. So for those of you who are interested in this, you can talk more. You can email me and we can discuss more about that. Although behold, I think this project, as you see from here, again in science, yeah, you're right, Joey, and science engineering, we are now seeing more and more women scientists, graduate students in the research project. And much of the work is done by Darren, who was a graduate student, subsequently become a postdoc in my lab. And this work would not be possible with the collaboration of many people in the team, and also my very exciting colleague from engineering, Tony Kanchanawang, and also Professor Roger Fu from School of Medicine. And this is just one of the very many exciting projects we have a chance to work with within the NUS. This is about the hub project. Now, before I go, I think please, there's a lot exciting program in NUS, and to say the most exciting nurturing environment, the support from the university and the exciting graduate student and postdoc really define who we are and what we do. And please go and visit a booth, and my colleagues at Science and NBI are very happy to enlighten you further. So thank you very much. Back for time, I take a couple of questions.